Hello everyone, my name is Asisipho Mbingeleli and welcome to my YouTube channel. In today's video, we are going to be looking at the full description for DNA replication straight from exam guidelines. Whenever you're asked to describe the process of DNA replication, it's normally 6 marks. So we're going to mention all the points that you need to know in their correct order in order for you to get 6 marks. So let us not waste time. The first point, the DNA double helix unwinds. The DNA double helix unwinds and the hydrogen bonds break. Now, you don't necessarily need to mention the word weak. We know it's weak hydrogen bonds, but you don't necessarily have to mention. Or you can talk about the molecule unzips. Okay, so for the second mark, you'll either get a mark for mentioning hydrogen bonds break or the molecule unzips to form two separate strands. Each strand serves as a template, or you can say both strands serve as templates. You can use the word both or each. Both strands serve as templates to build a complementary DNA strand. A complementary DNA strand. For that particular point, you can also say that we know that A pairs with T and C pairs with G. Okay. Then the next point. Now, in order for us to build the complementary DNA strands, we are going to be using free DNA nucleotides. This is very important to mention that we are using free DNA nucleotides. And where are these nucleotides from? They are from the nucleoplasm. Now, obviously, we are now going to have the template strands with the complementary DNA strands. Now, this will obviously result in two identical DNA molecules. Now, let's talk about how this whole process is marked and also some common mistakes that you need to avoid. Now, from the first point, you need to mention that you're having the DNA double helix molecule and that it unwinds. Very important. And the hydrogen bonds break, or you can just say the molecule unzips for the second point. For the third point, for third mark, to form two, you need to mention that there are two separate strands that are formed. Remember, the molecule unwinds and then it unzips, then we end up having two strands. These are the two strands, right? Then both strands, this word is very important, both strands, or you can say each strand. So you'll be marked for that. So both strands or each strand is going to serve as templates in order to build complementary strands. So what we're definitely having here, so remember the molecule unzips. So you're going to have template strand 1 and another template strand. Now we need to build complementary strands, and how we are going to build complementary strands, I'm going to use another color. These are strands that will form here in the middle. So this will be a complementary strand and this will be another complementary strand. We are going to use free DNA nucleotides. This word is important because you are telling us which nucleotides we're going to be using from the nucleoplasm, and these are the nucleotides that are going to pair with the existing nucleotides in the template strand. So for example, let me get a bit of space and then rewrite that. Okay, let's see. So I'm just going to say this is the template strand and this is another template strand, and this is another template strand, and the exposed bases for these strands. So let's say we have TAC. That means this side will have ATG. So these are our template strands, and we're going to use both of them because both of them are going to serve as templates. Now we're going to build complementary strands, and we build those strands by using the DNA nucleotides. Now I'm going to use a different color. So we're going to use we're going to build a complementary strand that will complement this template strand, and we're going to build another complementary strand that is going to complement this template strand. So let's look at this one. So A will be complemented by T, then here we'll have A, and here we're going to have C. Remember, we don't talk about T here because it's all about DNA, nothing to do with RNA. Okay, so this will be a complementary strand. So that's C on top there is for complementary strand. Then for this particular complementary strand, we need bases that are going to complement the exposed bases of this template strand. So T, that will be A, that will be T, and that will be G. I hope you can see what is happening. So then this will result in two identical DNA molecules. So where are these molecules that we are talking about? So this will be one molecule, and this will be another molecule. Now each molecule consists of a template strand and complementary, template strand and complementary strand. Now it's very important for me to mention that at the end we're having two identical molecules and not strands. You can't say you're having two identical strands because actually at the end there's 4 strands, but you can't say you're having 2 or 4 identical strands. No, you actually need to say DNA molecules. Here on point number 4, when you use the word one, that will be incorrect. So you can't say one strand serves as a template because that's a point for transcription. So please avoid that mistake. On this particular point, using free DNA nucleotides, please do not make a mistake of saying using free RNA nucleotides. And do not make a mistake of saying these nucleotides are from the cytoplasm. So cytoplasm will also be incorrect. I hope it all makes sense. Please remember to subscribe to the channel, guys. Hit that like button, share the link with your classmates and your schoolmates and your friends, and also please comment down below.